Tonight on Panorama, the scientific breakthrough that could change the lives of everyone and everything on the planet. It's an advance in gene editing, which holds out the promise of cures and personalized treatments for some of our most deadly diseases.
Imagine a tool that allows scientists to change the letter code in the DNA of a cell so precisely that we could change a single base pair in the 3 billion base pairs of the human cell.
It sounds complex, but gene editing has just been made simple and is revolutionizing research into life's big killers and the diseases of aging.
Gene editing has created a fundamentally new kind of medicine, and this means that we can now treat genetic disease, infectious disease, and cancer in ways that 10 years ago would have seemed like science fiction.
It crosses the animal and plant kingdoms and kickstarts a new era of genetically modified organisms. We can now control evolution so precisely that insects which spread disease could be eradicated. But medicine's big breakthrough is not without risk. I've set out to discover how gene editing could change our world. It's just 4 years since researchers discovered a new technique to edit DNA called CRISPR, which is so fast, cheap and accurate, it swept through nearly every field of scientific research. I've come to the west coast of the United States, to San Francisco, to meet a pioneer in this fast-moving field of science, one of the co-discoverers of CRISPR. The University of California, Berkeley.
The pace of science has just increased incredibly.
Jennifer Doudna is a biochemist and now one of the world's most influential scientists. She's tipped to share a Nobel Prize for discovering a new form of gene editing.
CRISPR is an acronym, and it stands for Clusters of Regularly Interspaced Short Palindromic Repeats. Big mouthful, easier to say CRISPR.
Don't let the terminology put you off. Put simply, the CRISPR system acts as a chemical cleaver which allows scientists